Hi students, welcome back to this video. See, here is an inspirational quote. Listen to this. You don't have to be great to start, but you have to start to be great. Yes, this inspirational quote can bring out the best from you if you execute it with the actions. Okay, students, come back to the topic. The topics to be covered in this session are regulation of light in the eye, formation of image, and retina and photoreceptors. Here, the first topic is the regulation of light in the eye. See students, the entry of a large amount of light is harmful to the tissue of the eye. Iris and the aperture, that is at the center, called pupil, regulate the amount of light falling on the eye. See, you analyze the picture or this photograph and try to find out how the eyeball regulate light actually in the intense light or bright light and dim light. What is the role of iris? This portion is known as the iris, and the middle, this aperture or this opening, that is known as pupil. How do these iris and pupil regulate the entry of light during dim light and in the intense light or bright light? Here you can see there are two types of muscles present in the iris. The name of the muscles are circular muscle and radial muscle. So these muscles are known as the circular muscle and these muscles are known as the radial muscles. You know, the dilation of the pupil is done when the radial muscles of iris are contracted. Means the dilation of the pupil takes place when the radial muscles are contracted. So here you can see a wide opening. This is dilated. Pupil is dilated so that more light can enter towards the eye. This picture is entirely different. You can see that here there is less opening so that less light only enters into the eye, which means pupil is constricted. Constriction of the pupil is taking place when circular muscles of iris contracted. These circular muscles, whenever it is contracted, that time constriction of pupil we can see, so that less amount of eye only enters into the eye. So from this picture, these two pictures, you try to understand in which all condition takes place. So the first picture takes place in which condition? My question is either in the dim light or in the bright light. Yes, of course, it is taking place in the bright light. People constricted because of the circular muscles contracted. Okay, here in the dim light, pupil is dilated because the radial muscles of iris contracted. So I'll give you another photograph to show this constriction and dilation of the pupil. So in bright light, pupil become smaller. See, This is the side view. And this is the front view. Front view, we can see the pupil becomes smaller. What about in the dim light? Pupil becomes larger. We can see here, it is very clear. This is the side view and this is the front view. The larger pupil we can see. Then one more photograph is here. Then it will help you to understand very clearly regarding the regulation of light in the eye. So students, listen carefully. Sorry. Okay, you can copy down the points because in dim light, what happens to the radial muscles? In dim light, the radial muscles contract, so that dilation of the pupil took place. In dim light, contraction of the radial muscles causes the dilation of pupil. In bright light, contraction of the circular muscles, these are known as the circular muscles, that helps the contraction of the pupil. Okay, I think it is very clear to you. Then see children, actually here some points I have written. From these points you will understand more about the regulation of eye. When the radial muscle contract in dim light, the size of the pupil increases. When the circular muscles contract in intense light, the size of the pupil decreases. Thus, the amount of light falling on the lens is regulated according to the intensity of light. Okay, then our next content is, what is the next content? Formation of image. Yes. How the images are formed in the retina. Especially, what about in the near object and distant objects? How do we see things which are present in the distant place as well as the nearest place? See, here the curvature of the cornea and the lens help light rays which reflect from an object to get focused on the retina. Similarly, we can see nearby and distant objects clearly. Yes, this is because the focal length of the lens can be adjusted according to the distance of the object. So according to the distance of the object, the focal length can be adjusted. So here, in the case of distant object, what all changes we can see? Especially shape of the lens as well as the focal length. See here, the curvature of the lens is decreased so that focal length is increased. Then curvature of the lens decreased means with the help of ciliary muscles and ligaments. So here what happened to the ciliary muscles when we see distant object? Ciliary muscles relax and ligaments stretch. Then I will show you where the ciliary muscles are present. See children, ciliary muscles, we can see ciliary muscles are present here. When the curvature of the lens increases and decreases, the ciliary muscles plays major role. Then this side we can see ligaments. Here we can see the ligaments. Ligaments are present here. So ciliary muscles and ligaments. Whenever the curvature of the lens decreases, ciliary muscles relax and ligaments stretch. Okay, curvature of the lens decreases, focal length increases. So write down these important points. How do we see near objects? How do we see distant objects? These are the points that I have shown you. See, while viewing distant objects, ciliary muscles relax, ligament stretch, curvature of lens decreases, focal length increases. So next is regarding how do we see near objects. See, the curvature of the lens increases and the focal length decreases. Then here also we can see ciliary muscles as well as ligaments. Ciliary muscles are present here. See, then we can see the ligaments. They are also present here. Then what all changes take place in the ciliary muscles and ligaments when we see the object which are present very near to us. So when we see or while viewing nearby objects, curvature of the lens increases, focal length decreases, ciliary muscles contract, and the ligaments relax. So these points will help you to understand very clearly. So listen here. These are the important points regarding while we view near objects. Ciliary muscles contract, ligaments relax, curvature of lens increase, and focal length decrease. Then our eye has the ability to adjust the focal length. So that is known as power of accommodation. So you listen here. The ability of the eye to adjust the focal length of the lens by changing its curvature in accordance to the distance of the object from the eye and form the image on the retina is called the power of accommodation of eye. From your examination point of view, it is very important. So you study what all changes takes place while viewing near object and distant objects. Then what do you mean by the power of accommodation? So the next is or next topic of today's session is retina and photoreceptors. So we can study what about the photoreceptors. Okay children, there is one more diagram is there to show you this view. How do we see the distant object and near object? See the diagram. See, out of this which one is found when we see distant object? Yes, the first one, because the curvature of the lens decreases and focal length increases. Okay, then retina and photoreceptors. Here, don't you remember where the retina present? What about the photoreceptors? And what do you mean by receptors? Yes, rewind it, all these things. Receptors are nothing but they are modified neurons, and photoreceptors means these receptors are stimulated in the presence of light because the word photo means light. Okay, then what about the retina? Retina is the innermost layer. Okay, then where these photoreceptors are present? Of course, the photoreceptors are present in the retina. What are the two types of photoreceptors? What is the significance of these photoreceptors? What about the chemistry of vision? How do these photoreceptors play major role for viewing objects? All these things we should know. Okay, so there are two types of photoreceptors. They are rod cells and cone cells. Then one more important point, the number of rod cells are more. In the retina, the number of rod cells are more. We should remember about this. Rod cells are more in number. There is a question regarding this statement. Then what is the function of rod cells? Actually, rod cells help us to see in dim light. Then rod cells consist of a pigment named rhodopsin. Then one important thing, rhodopsin cannot detect colors. Then how this rhodopsin formed? Rhodopsin is formed of two important substances named opsin and retinal. Opsin is a protein molecule, while retinal is derived from vitamin A. We all heard we should use more leafy vegetables. It contains more vitamin A so that you will have the power of sight more. Yes, it is because this retinal is formed from vitamin A. So what all things we should remember while talking about the rod cells? Yes, rod cells are the photoreceptors which is present in the retina that help us to see in dim light. Yes, next, they cannot detect colors and it is made up of pigment called rhodopsin. Rhodopsin consists of opsin and retinal. Opsin is the protein molecule while retinal is derived from vitamin A. Now you will understand we are unable to see colors during dim light. Why? Yes, it is because rhodopsin cannot detect colors. If we want to see the colors, yes, we need bright light. So that we are going to study. What about the cone cells? Yes, cone cells help us to see in bright light. Yes, cone cells help us to see in bright light. BRIGHT. Bright light or intense light. Then it also consists of retinal and opsin. Actually, the pigment which is present in the corn cell is known as, two names are there, photopsin and iodopsin. These are also made up of retinal and opsin. Opsin is a protein molecule and retinal is derived from vitamin A. The other important factor regarding corn cell is there are three types of corn cells in the eye that help us to detect primary colors such as blue, red, green. Yes, this diversity is due to the difference in amino acids in the protein molecule or opsin molecule. Yes, corn cells provide us color vision. Corn cells help us to see in bright light that we study. See children, what about the chemistry of vision? Uh, you understood corn cells and rod cells, how these photoreceptors help us to see in dim light and bright light. What about the pigments which are present in the rod cells, corn cells, etc. So next we have to study little more. We have to study more detail about the chemistry of vision. See, in the presence of light, rhodopsin, actually that is in dim light, it is dissociate into retinal and opsin. In the presence of bright light, this photopsin is dissociated into retinal and opsin. So this chemical change leads to the formation of impulses. Then it is transmitted to the cerebrum through optic nerves and enables vision. Sorry, children. So this chemical change, which chemical change? That means rhodopsin is converted into retinal and opsin, and photopsin is converted into retinal and opsin. Actually, that is taking place in the presence of light. So that chemical change leads to the formation of impulses. Then who carries these impulses to the cerebrum? Cerebrum can only can help us to see things. So cerebrum coordinate and gives us the perseverance sight. So here the chemical change leads to the formation of impulses. It is transmitted to the cerebrum through optic nerves and enables vision. See children, we have two eyes and in the two eyes two images are formed. But we can see an object as one only. Why? So that is known as the binocular vision, that ability. So the images from two sides of the same object are formed in the left and right eye. When these images combine as a result of the activities of the brain, a three-dimensional image of the object is formed. This is called binocular vision. Actually, this is done by the cerebrum. Cerebrum coordinate and gives us the only one image. Okay, students, when we summarize today's topic, yes, what are they? First one is regulation of eye. Second one is formation of image. And the third one is about the retina and photoreceptors. That's all about today's class. Thank you for watching this video. Thank you very much.